My task is sort of a dual task. I'm going to talk about this vaccine in a short period of time, and then I'll talk about the challenges ethically. So what has happened, it was touched on by Hemike also, that the changes in cancer treatment is quite radical. There's a paradigm shift, and by that it means that we usually have surgery, radiation, chemotherapy, and target therapies. Had that for a long time. But what's come during the last years is the fourth foot to stand on, is immunotherapy. So actually it's a paradigm shift. It has changed a way of thinking, also a way of doing clinical trials. So what we have seen is then is the normal the number of patients being alive and this time. And you see here that the controls, that means those were untreated patients or those who got chemotherapy, because in solid tumors it hasn't been that very successful, with few exceptions. But now we got immunotherapy. And what we see then with these kind of treatments, the survival curve flattening off. Is a new entity called tail of the curve. That means that patients continue to live, although the treatment was given maybe 3 or 4 years before, they still continue to live. And now combining different immunotherapies, we may even elevate the curve even more. And that's what we are doing right now. So there was a whole new world for melanoma. I've been working in melanoma field for many, many years and tested a huge number of different new drugs, but nothing worked. But now we got something new, which was this. This is a survival curve showing patient at risk, 4,800 patients. It's a meta-analysis of using a new drug called ipilimumab, which is a checkpoint inhibitor. And as you can see here, if I can see here, you see the survival curve tapers off, but then after 3 years, it sort of levels off. Now this is then 120 months, that means 10 years. That can be expanded maybe to 12 years or 13 years for a number of patients. And at our hospital, we started with this kind of treatment in trials 6-7 years ago, and we have patients that had treatment for 6 years ago. They still have something in the lung and the liver, but nothing is happening. So what we are allowed to say that we probably have cured most of these patients. And that's something we would not see possible for 3, 4, 5, 6 years ago. We're probably able to cure it. This is melanoma. It doesn't mean that it works for every kind of tumor, but it's something you have to take into account when you do clinical trials. It has been usually, as was said earlier on, prolonged the survival, maybe 2 or 3 months or so on. But now you are in a different setting. With these kind of treatments, you may cure patients. And that's quite different. So these things were so huge, the Nobel Prize was awarded to Honjo for one of these new checkpoint inhibitors, the anti-PD-1, and Jim Allison for his discovery of the anti-CTLA-4, which are the first 2 checkpoint inhibitors. But there are many more now coming on. New are being developed, and combinations of these checkpoints is our hope to even increase the effect of the treatment even more. But they don't work in everybody. I mean, the hallelujah, emphasis that all this is going to be great and everything's going to be solved this way with immunotherapy. Yes, something is solved. We have elevated the survival curves, but, it's a huge number of patients who have no benefit from this treatment. So we need to improve that therapy. And there are many ways of doing that. You can say that we could optimize the dose, because we don't know exactly what are the right doses, because these are biological treatments. And they are not like chemotherapy, because then it could be that the more you give, the better effect you have. But in biologicals, that might be quite different. So the doses we have chosen at the moment, they are sort of arbitrarily. It could be that other doses are more effective. And we do need biomarkers. That means we need markers to say which patient would benefit, who will be the one who will be part of that tail of the curve. But we don't have that. We're trying to find it, but as of yet, we don't have any good biomarker telling you that patient will have benefit, that patient will not have a benefit. We can combine new checkpoint inhibitors because there are many, there are huge numbers actually, and they might improve the effect of the treatment because, as I said, it doesn't work in all patients. It could be that we change from inhibiting the inhibitor, because the treatment we have used until now has been mostly. Is it on?
Is it on? It's on.
It's not enough. Is it better now? I have no idea. What also we have, what we're relying on with these checkpoint inhibitors is that inhibiting the inhibitors, because we know patients have immune responses to the tumor, but it's downregulated. So we take away is a downregulation of the immune system. That means that the patient's system has understood this is something bad, but it's not able to play out the whole army of T cells. But you could turn that around and say, well, we can not only take away the inhibition, we might also stimulate, and that is something we can do also. That's going on at the moment of trials. But yeah, and these are some of the ways is that blocking the inhibition, what we have been doing until now. You see here CTLA-4, PD-1, as I mentioned. You can use that. Others are coming. LAG-3, TIM-3, and so on are in the pipeline. So we'll see how good they work. The other way around is to turning up the activation, which is kind of touchy because you may do too much, and too much activation of the immune system could be dangerous and could give toxicities which can be quite serious. So this is ongoing. It's a tremendous number of clinical trials on this field. I think maybe a couple of thousand of trials, I think, are being tested. So it's a problem now that so many possibilities of combination that we don't really know what is going to be the best one. It's going to take time to find that out. But the one thing we think that the PD-1 ceiling that we have sort of reached at the moment, that's my opinion, we need to do something novel. Not only combining the checkpoint inhibitors with new ways of doing that, but we probably need a way of recruiting new T-cell clones. And doing that, we could do, as Emeka told about, we could use oncolytic virus, which will help you in that situation. You could do cancer vaccines, or you could do long peptide vaccines. I'm going to talk about the last one. So then something quite different. I don't know if you can see the red dots? Yeah. They are telomeres sitting on the end of chromosomes. And what they do is they protect the end of the chromosomes, because when the end of the chromosome have no more telomeres, which is the one adding on, that tumor, that cell will die. So actually there is a system in the body with biological clocks. So it means that if a normal cell are dividing, they may have a number of divisions to be performed. After that, the cell will die or go into a senescence. These tumor cells, though, they are smart guys. They have an enzyme, telomerase. So after each division or mitotic division, the telomere that got lost is being added on because they have an enzyme, telomerase. And that characteristic is typical for cancer cells. Because if you take cancer cells, you know, they can live in the lab forever. And why do they do that? Because the biological clock, it doesn't work, because they add on more telomeres every time they divide. So that's typical characteristic of a cancer cell. So this is just to show you the different way. A normal cell will, sorry about that. So, like this. Yeah. Well, kind of pointer is difficult to find. Yeah, here it is. So a normal cell would die after certain division, but the tumor cell will continue to live and divide because the telomeres will not be taken away. So, and that characteristic is something all tumor cells have, almost 90%. It's present also in the cancer stem cells. Is essential for unlimited growth and immortality, which is typical for cancer cells. And most normal cells are telomerase negative. So in a way, telomerase peptide vaccine, or targeting the cells expressing H or telomerase, which shows H30 on the surface of the cell, could be an ideal antigen in cancer therapy. And not rely on being it a colon cancer or a prostate cancer, but it could be a general vaccine. So we have done quite a lot of work with a peptide targeting cells with H30 on the surface. And this is on results from in 24 patients with lung cancer, stage 3 and 4, 3B and 4, inoperable ones. And we saw those patients who showing an immune response, they had a much better survival than those patients who did not show an immune response to the telomeres. So there's a correlation between immune response and overall survival. And those patients who did not respond had a poor prognosis, as you see here. So what we looked at then, we have patients who survived 10 years. Not that many, because the initial trials with the vaccine wasn't that good, because there were some patients who had fantastic effect, but there were too few. And the reason for that is probably that we did not know that there was down-regulation of the immune system. We didn't have the anti-CTLA-4. We didn't have the PD-1. But we have isolated the peptides, which these patients who survived more than 10 years, we know which they are, and we made a vaccine out of that, because those peptides probably, although would probably be a useful one for having a good activity in patients. So we have done clinical trials here. Here is the immune responses in patients with UV1, that's the vaccine, in prostate and in lung. And you see quite a substantial part of the patients show immune responses. But if you combine it with a checkpoint inhibitor, it comes more quickly, and almost 95% of the patients shows an immune reaction. So we thought this is something we could develop, it could be useful. So we have, and this is then combining, this is in malignant melanoma patients. These are the patients who received ipilimumab, the anti-CTLA-4 alone. And these are the 12 patients, mind you, 12 patients is not very much, compared 12 to 70. You could criticize that all the time. But it's astonishing that you may have a leveling off of the curve. So it was encouraging, but proving nothing. We have to do clinical trials at a larger scale. And that is something we're doing now. This is then the UV1 vaccine combined with another checkpoint inhibitor, which is the later one, the anti-PD-1. And that is ongoing in the United States at the moment. 5 sites, and as a phase 1, we are looking into endpoints like safety, immune response, and efficacy. And this is ongoing at the moment. 6, 7 patients are now being included. No major toxicity as of yet. But next step, this is the phase 1, it's just the start of a long development of a drug, is a randomized trial, here down at the bottom. Phase 2 trial randomized with ipilimumab and UV1 vaccine, giving the 2 checkpoint inhibitors together with the vaccine, and the other arm is without. And that's going to be in Europe and in the United States. We hope to start that in late this year. So this is the development in the company called Ultimovacs, which I give advice to. So, ethical changes. That was the second part of my talk. The thing is that ethical challenges in these trials, which I mentioned now, well, these patients get an already established treatment, which means that they have a chance of efficacy. The question is then, what kind of toxicities may they have? Well, vaccines normally do not give that much toxicity, but we have to remember that what we do, we induce T cells, which will react to cells which show H-church on the surface, and that might be forever. We don't know if that can do something in the late, maybe 10, 15 years. We have maybe now 10, 12, 13 years observation. Haven't seen any bad effect of that, but one should keep that in mind. But generally, one can ask always, is it really ethical to offer trial participation in patients who had life-ending illness? And that's something we discuss all the time. Because whatever you do, you take away valuable time spent in hospitals, frequent blood sampling, X-ray examination, travel, and all that. And sometimes, that time could probably, in some extent at least, and when the treatment doesn't work though, could be spent with family and values which are maybe more important for those at least left behind. So that is something we have to ask our trials all the time. And going into a clinical trial, we have no toxicity and no anti-cancer activity is really challenging. Another question is that, are these patients really able to make a voluntary decision? on trial participation? Is that really fair? Do they understand the patient information or patient consent form? And do the patient have equal access to these clinical trials? And that we discussed that earlier on, the privilege and underprivilege, they are not, I'd say that. Our experience is that mostly it is patients well-educated, good developed networks and so on, are the one who will look for phase 1 trials when nothing else works. So we'll be talking about, well, we talk about risk-benefit. It was touched on early on. And that is important. But all kinds of clinical trials, we have to rely on that. This is for the patient themselves, but also for the society. There is should also look into that, because there are huge numbers of drugs. I did animal work for my 6 years. I don't know, 1,000 of mice. Did a wonderful treatment for mouse cancer, but a lot of those, most of them, do not work in patients. A lot of common with mice, more than you might like, genetically, and also you can open a mouse, everything is there, but they are different. Useful model, but I've lost a little bit of my confidence in animal studies, certainly for immunotherapy. So what happens then with these patients? Well, this is a study done in 11,000 or 12,000 cancer patients. NCI came out in 2005. And you can look here, what are the results? I don't know if you can read that. But it's chemotherapy, it's overall response rate of 10.6, CR 3.17.5. Rather high, actually. But phase 1 trial patients are not the common patient in many ways. They are better health sometimes, and they feel like I want something more of the treatment, which may mean that they are in good condition also with their cancer. So I think these response rates are rather high. And you could go down the list here in different kinds of treatment, vaccines, gene therapy. It is a response. If that's enough, that's something, I don't know. But to say entirely that phase 1 trials do not have any activity on the tumor is not correct in my opinion. What happens then? What about mortality? Because when you take a drug from mice, you do some toxicity studies. You know exactly what happened with mice at that dose level. When you start out with it in a phase 1, you start at very low because the toxicity may be quite different in humans than in mice. And you probably would expect that some patients may die from the toxicities, and they certainly will. But I was surprised, maybe 1 or 2%, maybe 1% deaths caused by toxicity. So it's not that risky, as one would think, because we sort of start at a very low dose and then climb up till we see some signs of toxicity. So my opinion, it's not that unsafe. This is from Royal Marsden. They also show that Judson did an analysis in 2008, and they found that there's clinical benefit in many ways for patients participating in clinical trials. Now, how many of these drugs who entered the development line, which was shown early on, phase 1, phase 2, phase 3, and finally then registration, will succeed? And at the bottom here, as you said, there was 1.7% ended up as a drug at that time. But that is improving. And why is that so? Well, we have biomarkers which might help us to select the patients who have the more likelihood of participating. So I think this is going to continue to rise with more drugs getting to final approval and with a higher probability of success. But still, it's, maybe it's going to be 8, 9, 10, 15%, but not more than that, for sure. Another question is, who should decide on the risk-benefit? When is it really the risk-benefit okay to start a clinical trial? Should that be the investigators? Bioethicists, you guys? Lawyers? Dietitians, physicians, policymakers? I'm not quite sure about that. Or should people who are facing this have a life-ending illness and who will have the toxicities and all the hassle it is to be kind of a part of a clinical trial, maybe they should be participating and deciding what is useful. So patient participation is very important at early stages. It hasn't been the rule, actually. And the reason for that is that patients with a serious disease have a different perception as healthy people. So patients have, when you're in that, maybe on the edge of the cliff, you may think differently than you are a healthy person in society, active in your life, and you have a job and everything. And that's something I discuss also, criticize the use of QALY, quality adjusted life years, which is used to evaluate the value of a new drug treatment, which is, in my opinion, totally false, because those values, sitting in a wheelchair, is half life quality. That's not true. I mean, it's not true. It was done by healthy individuals, and that's unfair. We should look at the patient who really are part of the situation, and they can tell us what that life is worth. And sometimes the family consistently overestimate symptoms and underestimate the patient satisfaction and quality of life. So we have seen many times the patient actually lead a small benefit to find cancer chemotherapy, which we talk about here, worthwhile. They were asked in a survey, nurses need a 50% chance of having a benefit of a treatment. Doctors a little bit more skeptic, 10%. General public, maybe 50%. But cancer patients, sometimes say, well, 5%. I have been asking patients, the risk, what will you accept? 1%, well, I'm going to be the one. So, I mean, that's a way of looking at it. So, and this is then we're asking patients, what would you do if you got hair loss, nausea, fatigue, 10% chance of death? Would you then not participate? No, they say, no, I would still participate. So again, that is something asking the patient, not the physicians and not the healthy individuals. So patients say, perhaps this will help someone else. This may not work, but I can cope with doing nothing. I can't cope. I'm just sitting there and waiting to die. This is my last chance. My consultant told me there was nothing more to do than phase 1 if I wanted some treatment. I might give me a bit longer. Patients are willing to bargain for months almost, maybe reaching that certain date when something is important happening in the family. Or the really optimistic, I'm aiming for a cure. So one has to think about the difference from hope and expectations. It's so difficult. So one talk about therapeutic optimist, who's the one who thinks that this is going to work much better than these guys tell me. And also maintaining hope is important. We should not treat, as you said, early on for giving patients hope. That's not fair, because they will certainly have toxicities from the treatment. But one should not underestimate hope and should not take away all the hope from patients. That's very important as an oncologist, because hope may be living some time, maybe without pain for a while, and so on. And for some, it's a coping strategy for patients. But what can they have a benefit? Am I over time already? Okay. Because there are a direct benefit, of course, when the tumor is shrinking, maybe even go away. That happens with immunotherapy at the moment. But there are indirect benefits also, that being part of a trial is good for patients, although that cannot be the aim. That quality of life improves when you have general good contact with your doctors and your nurses. And patients in clinical trials, you have a fantastic situation. They may have a business card. They have called my nurse when I don't feel well, which is not happening in the healthcare system at all. And also some comfort, that helping future patients, or maybe it's my grandchild who may benefit from what I do right now, because I'm participating in the drug development. So I think over time, I will not go into other things, but sometimes I would say, patients in this situation cannot make any good decisions because they are so on the edge of the cliff. It's unfair in a stress situation. I don't think that is true. I mean, a lot of patients have absolutely ability to decide on not participating, and they also understand quite well the toxicities and also the possible, the impossible sometimes effect of the treatment. So it's pressure was talked about, family could pressure patient into clinical trial. But, the main thing is that, is growing cancer is the motivation for patient participating in clinical trials. But equal access, I have to skip that. I think that's important. It's very important, actually. As we said was initially, in early phases, we see that patients, we are well connected, well educated. The privileged group of patients, also in Scandinavia, is like that, are the majority of the patients participating. It shouldn't be like that. It should be for everybody. So, okay, last one. Yeah, it's they are driven patients by an urge for survival, and for this reason they require protection. So we have to be very accurate in what we do. We have to do all the things with transparency, information all the way, and the uncertainty which a clinical trial carry. And remember also patients are willing to accept maybe 5% chance of, or less even, I've seen 1%. And we need to ensure equal access. But while we do that, we have to remember that it's the only means we can improve patient care. Because we're too negative in participating in clinical trials, how will we develop drugs? There are no way of doing it. There are no good models. There are no animal models. People say, well, could you do in monkeys? No, you can't do that in monkeys. You have to move it from the lab to the patient. Thank you.